Good afternoon. I am excited and honored to introduce the second Max Harry Weil Honorary Lecturer, Dr. Polly Matzinger. To say that Dr. Matzinger is unconventional is an understatement. She has had numerous life experiences and jobs before she began her innovative scientific career. While it's unusual for an NIH section head to have a web biography on the Internet Movie Database, in Dr. Matzinger's case, that entry provides an excellent summary of her transition into science. Let me share a quote from that site. Matzinger came up with a breakthrough in immunology while spending an afternoon with her border collie, who was awakened by noises of fear from a nearby flock of sheep. Using the event as a metaphor, she realized that the immune system is like a collie who looks for signs of distress in the body. She subsequently received her PhD at the University of California, San Diego, with postdoctoral training at Cambridge. She is now a tenured researcher at the National Institutes of Allergy and Infectious Disease, and she is the head of the T-cell tolerance and memory section of the Cellular and Molecular Immunology Division. While many of us question ourselves as we grow, her fundamental questions about what self means to the immune system is the foundation of her work. Her theories and investigations on danger and damage signals have markedly altered our viewpoint of immunology. Her perspectives are highly relevant to the clinical problems that we encounter on a daily basis in the ICU, and her specific topic for today is a new way of looking at immunity and injury. Please join me in welcoming Dr. Polly Matzinger.
Thank you. Okay. Whoa. I was told you should never blow into a microphone, but I guess you shouldn't tap it too hard either. Wow, I can't see you, which means that if I get confusing, I'm not going to know it. I have to start with a question. How many of you are not immunologists? Okay. How many of you that are or are not but had an immunology class were taught that the immune system functions by discriminating between self and non-self, and that it does this by learning self early in life? Anything that's around early is called self. Anything that appears later is called non-self. You leave alone self and you fight non-self. How many of you were taught that? I was taught that in the mid-'70s, and I have to say I had a terrible time with it, because my first question from my professors was, hang on a minute, sitting here today, we are not the same selves that we were when we were in utero or within a few days of birth, which is when we think the immune system learns what self is. And my professors patted me on the head and said, oh, there, little girl. Nothing new appears later in life, like in puberty and so on. It's just changes of concentrations of hormones and things that were already there. And so I pointed out a condition to them that they hadn't thought about, mostly because they had Y chromosomes. And that is this tissue. The newly lactating breast has cells that make proteins that body never made before, milk proteins. And even though most of them go out into the outside world, there's a few, like alpha-lactalbumin and beta-lactoglobulin, that actually end up in the bloodstream. Now, if this body was calling everything early in life self and everything that appears later non-self, she ought to reject this newly lactating breast. And, we don't. And so my professor said, that's because when she was learning self, when her immune system was learning self, she was drinking mother's milk and becoming orally tolerant. And so I pointed out to them that my generation's actually done an experiment. When I was born, it wasn't socially cool to drink mother's milk. You drank Nestle's or Carnation or pet milk. And these companies are still trying to do this to third world women. But the important point here is that when my generation grew up and started lactating, our breasts didn't fall off. Somehow the immune system knows how to deal with a changing self. That was one of the problems I had. There were lots of others. Among them, why don't tumors get rejected? Because we know that many tumors have new or mutated proteins, either virally encoded or mutated, or proteins that were on very early in development and never later. Why is it that when you need to make a vaccine, you need to add something like aluminum hydroxide? Otherwise, the immune system doesn't respond. There were lots of questions. And as a graduate student, there was no support in asking them and zero support for trying to answer them. And so I quit. And for the next 20 years, I tried to study how the immune system does it. How does it discriminate self from non-self? And after 20 years of trying and not figuring it out, I got a job at the NIH. And in the lab next to me, there was a young postdoc, an oncologist named Ephraim Fuchs. And Ephraim was asking the same questions because of the tumors. And unlike me, he wouldn't let go. So we spent 2 years arguing. And at the end of 2 years, we decided that an evolutionarily functional immune system is not one that fights everything in the air you breathe and everything in the food you eat. It doesn't make an immune response to the fetuses you carry or the sperm that gets you those fetuses. A useful immune system is an immune system that needs to fight things that are dangerous. And Ephraim said, a virus that comes in and makes 3 copies of itself and leaves without doing any damage isn't dangerous, and you don't need to make an immune response. So that was fine. It was a cute idea. It took us another year to figure out, how does the immune system define danger? There are lots of ways of defining danger. Tumors are dangerous, for example, but the immune system doesn't usually reject them. And we came up with the idea that something is dangerous if it does damage, and it's not dangerous if it doesn't do damage. Now, that's a pretty simple idea. I don't know why it took us a year, but it did. And it took us another year once we got that idea to figure out the answer to the question of, if something has come into the body and is doing damage, how does the immune system know it? And what we came up with was the idea that the damaged cells tell the immune system. Now, when I talk to grade school kids, I try to put the old theory and the new theory in terms they can understand. And here is how I tell them. The old theory went like this. If the body is a community, the immune system are the police, the white blood cells. They're constantly going around looking for foreigners. And they define a foreigner as anybody they hadn't already met when they finished high school. And when they meet a foreigner, they kill them. That was the old idea. This kind of a community has no tourists, no immigrants, no traveling salesmen. The new idea, the danger model, says if the body is a community, the immune system is more like firemen. They sit in their little fire stations playing cards until somebody rings an alarm. And it's only when the alarm is rung that the firemen do something. And it doesn't matter if the alarm is rung by a local, a traveling salesman, an immigrant, or a tourist, they will do something. And unlike the police, they have more than one mode of action. They have a different answer if there's a cat up a tree than if there's a fire. And the danger model of the immune system works like that. You don't get an immune response, it says, the basic assumption, until a damaged cell sends an alarm. Now, when do cells send alarms? Well, not when they're healthy, not when they're growing, and not when they die by programmed cell death. There's a lot of death in the body. There's death in the bone marrow, death in the thymus, red cells are dying all the time, neutrophils are dying all the time, platelets are dying all the time. That's programmed cell death, and there are no alarm signals that go out. But if a cell is injured or stressed or dies by what I call bad death and some people call necrotic death, then you get an alarm. And the alarm is read by local dendritic cells. Every tissue that's been studied has intermingled with the tissue cells a population of immune cells called dendritic cells. And they're kind of like sleeping sheepdogs. They're asleep, noses under their tails, doing nothing. In the skin, they're called Langerhans cells, and each one is in contact with about 50 to 500 skin cells. They're asleep. But if a cell should send an alarm signal, what's going to happen is that that alarm signal from the damaged cell goes out and is read by a sleeping dendritic cell. And what that dendritic cell does when it gets that alarm, it has receptors for the alarms, is it wakes up and it starts drinking its environment. It can drink its volume every hour. It sends the water out, keeps all the solid bits, breaks them up into little pieces, and displays them on the surface in special Schlepper molecules that we call major histocompatibility molecules. So what you get are little peptides on the surface of the cell that are representative of what that cell has picked up. In the meantime, it traffics to a local node, and there it presents what it's picked up to the T cells that are going by. And when it presents them, 1 in a million or so of the T cells will see one of these peptides. Let's say this is flu. This is a T cell that can see flu. It gets what we call a signal 1 from that recognition event, and it gets a second signal from co-stimulatory molecules that are either secreted or on the surface of the activated dendritic cell. Now, what the danger model says is you never get these co-stimulatory molecules expressed by a dendritic cell unless it has first been woken up by an alarm signal. So the assumption is if you don't get damage and you don't get alarm signals, you don't get a primary immune response. It's a very simple assumption. It makes immunology really easy. And it's surprisingly potent assumption, because if you make this assumption that you don't get a primary immune response unless you have damage that activates a dendritic cell, you can explain almost everything the immune system does right and almost everything it seems to get wrong. So you can explain why you don't kill yourself at puberty, why lactation is not a problem, why mothers don't reject their fetuses, why you do usually reject transplants and don't usually reject tumors, why you get graft versus host disease, why there are parasites like filaria that live in the lymphatics in intimate contact with the immune system, and yet you don't get an immune response. You can explain autoimmunity. And what the model couldn't explain until very recently was allergy and asthma. And that didn't bother me. And the reason it didn't bother me is that allergy and asthma are a problem in a different universe of question. So the danger model was designed to deal with the first question the immune system has to answer when faced with something, and that is, do I respond or not? The old self-not-self model said you respond if what you're looking at is foreign. The danger model says you respond if what you're looking at is doing damage. Once you respond, you have a second question to answer, which is, what kind of response am I going to make? How do I know that I need to make IgE to catch a worm or killer cells to catch a virus? And allergy is a problem with that second question. Allergens are not innocuous. The main allergen in house dust mite, DERP1, is a protease that attacks the surface of lung epithelium and also of B cells. Bee venom is not innocuous. So what the danger model predicts is that every allergen is either causing damage, packaged with something that's causing damage, or mimicking one of the alarm signals. So I don't have time to go through all of this, but what I'm going to do is take 2 transplants as illustrations of how the model works. Mothers don't reject fetuses, and yet we do reject kidneys from a fetal donor. And then I'm going to go into what interests you the most, which is, why does the immune system do the bad things it does to your patients? Okay, so let's start with mothers and babies. The fact that mothers don't reject their fetuses was the first problem the self-non-self model had. If you fight everything that appears later in life that wasn't around early, you ought to reject your fetuses. And Peter Medawar, who got the Nobel Prize for doing the experiment that put that model on the map, realized this as early as 1953. And in his inimitable Oxford-educated British arrogance, it never occurred to him that the model might be wrong. He suggested instead that there was something wrong with female physiology. He said, fetuses immunosuppress their mothers. Now, that's really wise. You take the individuals that are carrying the next generation and you turn them into AIDS patients. And not only is it dumb, it's not true. How many of you have carried fetuses? Did you have to live in a bubble? No. Has anybody noticed? This stuff is still written in textbooks. Fetuses immunosuppress their mothers. Amazing. Now, it's true that there are some parasites like malaria that take advantage of the pregnant state. And that has been used as evidence that pregnancy is immunosuppressive. But it turns out, when people looked at the details, God is in the details, that when people looked at the details, that there's a malaria variant which makes a molecule that binds the glycocalyx of the placenta. The malaria and the fetus are really both parasites fighting for the same maternal resource, which is blood. And the fetus has had evolutionary time to figure out how to get it, which is to create a placenta, and the malaria goes to the placenta and steals it all. And so that's not good for the mother or the fetus. But by the second pregnancy, that mother is making an immune response, and by the third, she doesn't have a problem. Proof that she makes immune responses during pregnancy. Okay, so Peter Medawar didn't only say that. He said another kind of dumb thing. He said that the placenta is both a physical and immunological barrier between mother and fetus. Now that's great. That means if your fetus gets an infection, you can't do anything about it and you're dead. So not smart, and turns out, not true. There's traffic in both directions across the placenta. There are people, this started in Stanford, but it's spread around the country now. There are people bleeding pregnant women and isolating the fetal cells that are trafficking there as a way of getting tissue for a non-invasive prenatal diagnosis, which is there's traffic from fetus to mother. There's also traffic from mother to fetus. 25% of us sitting in this room are microchimeric for our maternal hematopoietic cells. This was first discovered in Leiden, in the Netherlands at Eurotransplant, when they started monitoring the immune responses of people who needed kidneys to their donors. When you have a living kidney donor, you test the recipient's immune response first, because you want to make sure that it isn't pre-primed, because that would be a waste of a kidney. It would get rejected really fast. Mothers are often donors. And it turned out that when they examined the immune responses of their potential recipients, 25% of them didn't respond to the non-inherited antigens on their mother's cells. They responded to their fathers, but not their mothers. And Lee Zhang and Rick Miller followed that up in Toronto with mice, where you can mate genetically marked mice and follow the cells. And they found that 25% of adult mice are carrying hematopoietic cells from their mothers. 50% of human skids, the boys who do have to live in the bubble, are carrying T cells from their mothers. So there's traffic from fetus to mother, and there's immune traffic from mother to fetus, and yet the mother doesn't reject the fetus. Why not? So the danger model says it's really simple. Fetuses don't look dangerous. The death that goes on in fetal life is programmed cell death. When you want to make a hand out of the primitive paw, you kill the cells in between. It's a signal the cells commit suicide. They don't spew their innards. They get scavenged by their neighbors, and no alarm signals are released. And so even though the fetus is foreign, it's not releasing alarm signals, so it doesn't trigger an immune response. Now, I know that at least somewhere in the audience, there's somebody thinking, what about Rh disease? You're not allowed to ask questions, I've been told. So I get to say anything. Anyway, Rh. So Rh negative mothers respond to their Rh positive fetuses. And this seems like a disproof of the model. But again, God is in the details. The detail is that the first pregnancy isn't usually a problem. It's the second pregnancy. Mothers get immunized at birth. Birth is dangerous. You have ripping, bleeding, surgeons with knives. It's a dangerous period. And we now prevent them from getting immunized by putting in antibody to block that immunization, because we know it happens at birth. All right, so the model says the reason you don't reject your fetuses is they're not sending alarm signals. If they get an infection, they will send alarm signals. And the immune system can go in and clear the infection. And if in the process of clearing the infection, you clear the fetus, at least you're alive to have another one. And we know that spontaneous abortion, spontaneous miscarriage, is a common concomitant of infection during pregnancy. Okay, so if we don't reject fetuses because they don't look dangerous, why do we reject transplants? Where's the danger in a transplant? So, I don't know how many of you are surgeons. The danger is the surgeon. Surgeons are dangerous. They cut, they burn small vessels, they put organs on helicopters and fly them around the country. They get death injury and reperfusion injury. That is not programmed cell death. Those cells are sending alarm signals. And so the prediction is that if you could do a transplant without doing damage, it should not be rejected. Now, you can't do a transplant without doing damage. Even a blood transfusion. You take the blood out, you cool it. That's called a heat shock. That causes the release of alarm signals. Heat shock proteins are alarm signals. However, Colin Anderson, when he was in my lab, did the following experiment. He took an immunodeficient mouse, rag knockout mouse. This is a mouse that does have to live in a bubble because it doesn't have a functioning immune system. And under the kidney capsule, he planted a neonatal heart. And the heart differed enough from the recipient that if this recipient had been competent, it would have rejected that heart in 14 to 26 days. But it's not competent, so it doesn't reject. And what he did is he left the heart to sit there for 9 months to heal. Why 9 months? Because it turns out healing takes a long time. He had done these experiments before with skin, left the skin for 5 months, and discovered that at the end of 5 months, it's still rejected. And that there are remodeling and repair enzymes still active. So he left his hearts for 9 months to heal. I now think that a transplanted tissue is not completely healed until it becomes reinnervated. But I have no data for that. That's just hand waving. People sometimes say, don't my wrists get tired of hand waving? Yes. All right, he left them for 9 months, and then he gave them back an immune system. And he asked, if now, after 9 months of healing, the immune system can see this heart, will it reject it? And the answer was no, and I'll show you that data. It's the only data I'll show you. They didn't reject. And he now asked a second question, because this is the other side of the danger model. The danger model says that if a new protein or other structure appears in the absence of damage or alarm signals, your immune system will become tolerant. And so what he did is he left the immune system and the heart to interact with each other for 5 months. And he asked, was the mouse tolerant or not? And the way he asked that was he now immunized the mouse against the donor antigens and tested for killing. And here are the data. This is an early experiment. It's 3 mice per group. And what you see here are 3 mice that just sat in the cages. They never got a heart. They never got a chance to become tolerant or to reject anything. They just sat for the whole time and were immunized. And then they were tested for killing. And what we do is we titrate the killers and look for an increase in kill. And what you see is that 2 of the mice are making phenomenal killers. And they're specific for the donor because they don't kill self. You probably can't see that, but there's another line here. And 1 of them is making really good killers. Here you have 3 mice that had had their hearts for 1 month before they got an immune system. 1 month is not enough. Even though the hearts are present when the immune system is growing up in that mouse, it's still sending alarm signals, and they were rejected. And we didn't even have to immunize. These are killers that come up just from the rejection immunization. And here you have 3 mice who had their hearts for 9 months before they got an immune system. And they didn't reject. And not only did they not reject, when the heart interacted with the immune system for another 5 months, it induced tolerance. So what this says is, if you could block the alarm signals, you could get transplants to take, and you wouldn't have to give people immunosuppressive drugs for the rest of their lives. Now, the alarm signals are going to be really hard to block. People have not found them all yet, but people have started. And here's at least a partial list. Anything that's inside a cell that is not normally outside a cell can be an alarm signal as long as the dendritic cells have a receptor for it. DNA is an alarm signal, RNA is an alarm signal, heat shock protein 60, 70, and GP96, they're all heat shock proteins, are alarm signals. ATP is an alarm signal. Uric acid crystals is an alarm signal. HMGB1, which is a transcription factor, normally found in nuclei and cytoplasm, is an alarm signal. And my favorite is mitochondria. There's a trauma surgeon at Harvard named Hauser who has been asking a question about trauma patients. And I didn't know this, but I learned from him that people who have really major trauma, you know this, I don't have to tell you this, people who have really major trauma, a lot of them die, and they die of what looks like sepsis, but you can't find an infection. And after hearing about danger, he started thinking, well, maybe there's something inside the body that looks like an infection. And mitochondria are evolutionarily ancient bacteria. And it turns out that mitochondria are amazing alarm signals. And what Hauser's shown, he's got a paper that's going to come out in Nature in the next couple months, is that when you make major tissue damage, the mitochondria that are released from the damaged cells, the mitochondrial debris, turns on the immune system. And when you have a lot of damage, you have a lot of mitochondria, and the immune system responds as though it were seeing bacteria. But it's not seeing bacteria. It's seeing an endogenous alarm signal. And he's shown, at least in rats, that he can mimic the disease by injecting mitochondria, and he can block it with antibodies to the FMLP receptor on neutrophils. So for those of you who care, you can ask me about that, more of that later. Mitochondria are my favorites. By the way, this clock didn't go off. How much time do I have left?
15.
Thanks. Okay, so you don't reject fetuses because they don't look dangerous. You do reject transplants because they look dangerous. And we can't block all the alarm signals, first because there are too many, and second because we don't know them all yet. So what can we do? What we can do is we can go one step up the path. Remember that I said what happens when you have a damaged tissue and you release DNA, RNA, ATP, et cetera, the dendritic cell has receptors for that. They're known as toll-like receptors because they mimic something in flies. But there are a lot of those, and you can't block all those either. But what you can do is to block the signal that the dendritic cell gives to the T cells, the co-stimulatory signals. There's a bottleneck in this pathway. There are a lot of different alarm signals and a lot of different alarm signal receptors, but only a small number of co-stimulators. And if we can block those, we should be able to get transplants to take. Now, there's a guy named Alan Kirk, who's a transplant surgeon, who actually tested that idea in monkeys. What he did was to transplant kidneys and to block one of the 3 known co-stimulator molecules. And he used an antibody to block that. He blocked it for 5 months and stopped. Now, he'd worked against himself in that he made transplant combinations where he knew the recipient was already making a strong immune response to the donor. And he blocked the co-stimulatory signal. And what happened when he stopped the drug is the kidneys were not rejected. And one of those monkeys is now at least 8 years out, having had no rejection episodes since they stopped the drug. Okay. That was in Nature Medicine about 4 years ago. If you can use blocking molecules like that to stop the rejection of transplants, we ought to be able to use them also to stop the sterile sepsis response that happens to critical care patients. Now, the problem with doing that, Kirk himself wanted to continue this work and do a clinical trial where he blocked all 3 co-stimulators because these monkeys still made some antibody and there was still some chronic rejection going. He wanted to block all 3. The problem is the 3 different drugs are made by 3 different companies, and he could not get the 3 companies to agree to get together on one clinical trial. So what he's doing now is he's making his own, but it takes a while. But let me tell you about another, again, a transplant result. There's an enzyme called superoxide dismutase. And what it does is it eats up free radicals, oxygen radicals. Oxygen radicals do damage. And in 1989, Walter Land, who was then the head of experimental surgery in Munich, did a clinical trial with kidney patients. And what he was trying to do was to increase immediate kidney function. Because in some patients you put a kidney in and it never works. It's not that it gets rejected, it just never works. And he thought it might be the oxygen radicals that occur upon reperfusion that causes the damage that prevents the kidney from working. And so he gave 44 patients 1 shot of 200 milligrams of SOD at the time of transplant, and another 44 patients not. And it didn't make a big difference for immediate kidney function. But 4 years later, the Institute computer nerd came running into Land's office. Professor Land, look at this, look at this. And it turned out that the 44 patients who had had the single shot of SOD had had almost no rejection episodes. Now, this was 93. And Land started thinking, what can possibly be the correlation between damage and rejection? And he told me later, you know, he's just a stupid surgeon, nobody would listen to him. I published in 94. In 96, somebody put my paper on his desk, and he started going around saying, look, even an immunologist, look, an immunologist is saying that danger and damage and immunity are correlated. He wanted to continue to do trials with SOD, and what he ran into is the problem that SOD is cheap. And the company that had the patent when he was doing his first clinical trial had either sold it to or bought it from Chiron, I don't know which way it went, and shelved it because they didn't want to spend the money for the clinical trial because they were never going to get their money back. Now, there's a lot of that, and as a basic scientist, I see it. And I think that it's a conversation we need to start having as a profession. Somehow we need to fund the clinical trials for things that are cheap. Maybe the insurance companies should be funding it because it's in their interests. Maybe the government should be funding it because I guess eventually we're going to have an interest in that as well. SOD, superoxide dismutase. If it helps people not reject kidneys by blocking the damage from oxygen radicals, it also might help other people not have immune responses to damage that is also going to be created by oxygen radicals. Okay. So the last 5 minutes? 5 minutes? It's hard. That says 9. In the CME credit thing, it said that one of the things you would learn about is autoimmunity. So I know that most of you are probably not interested in autoimmunity, but I think that autoimmunity is something that you may later become interested in, those of you who are not yet, because some autoimmune diseases, at least in my book, are created by damage. So I think we've been asking the wrong question about autoimmunity for the last 30 years. We've been asking the question, what breaks tolerance? Why does an immune system that ought to be tolerant of a tissue because that tissue was around at birth start making an immune response? I think that's the wrong question. And because it's the wrong question, we actually haven't gotten very far at answering anything about autoimmunity. So I think autoimmunity can be broken down into 5 categories, and in 4 of them there is nothing wrong with the immune response. Now, let's do them quickly. I only made up one of these categories. The other ones other people made up, but I've modified them a little. The first one is dead boring. You have an infection and you don't know it. I don't know how many autoimmune diseases are in that category, but I think the autoimmune diseases that flare have a good chance of being in there. My favorite example is Lyme. Lyme's disease was first diagnosed as juvenile rheumatoid arthritis until a nurse noticed that the kids that were getting it tended to live near the woods. And the CDC sent a young doctor up there to see what was going on, and he kind of stole her observation. How many doctors know the addresses of their patients? Anyway, she noticed that they'd lived near the woods. That started a hunt, and now we know that it's actually a parasite. It's Borrelia. It's not autoimmune disease. How many other autoimmune diseases are like that? The immune system is doing its job here. It's fighting an infection. Remember that ulcers for a while were thought to be caused by stress, and surgeons took out pieces of people's stomachs. And then Barry Marshall in Australia showed that it was actually due to a bacterium. He had to eat the bacterium, give himself ulcers, and cure himself with antibiotics before anybody believed him. But it changed medicine. Okay, the second category is similar but subtly different. It's called molecular mimicry. And mimicry, what it is a cross-reaction with an environmental stress, toxin, or pathogen. And my favorite disease here is actually not called autoimmune disease, it's rheumatic fever. So most of us, we get strep, we clear it without a problem. Some people, when they get strep, have an immune response that cross-reacts on the heart, usually the heart valve. Patients like this, physicians who take care of them tell me that patients like this are treated by giving them a ton of penicillin. Get rid of the strep as fast as you can, because if you get rid of the strep, the autoimmune response also goes away. So how many other diseases are like that, where we have an infection somewhere and a cross-reaction? What the immune system is doing here, again, is fighting a pathogen. The difference between category 1 and category 2 is that in category 1, like Lyme disease, there's no autoimmune component. So the infection has to be in the target organ. But in category 2, there is a cross-reactive component. You could have an infection in the gut that cross-reacts on the brain. Now, every time you clear that infection, you're going to do a little brain damage. But just because you cleared it doesn't mean it leaves your environment. You know, the next time you get on an airplane, the next time your kid comes home from school, the next time you go shopping, you're going to get it again. You will clear it fast enough that you don't notice it, but you'll do a little more damage to the brain. So the difference between this category and category 1 is that the infection need not be in the target organ. The third category I did make up, and that is bad death. And the disease I think fits in here is lupus, and probably others. So I think lupus is probably at least 300 diseases. What the danger model says is that good death doesn't get you an immune response and bad death does. And death, programmed cell death, is governed by a lot of genes. People that study it in the organism where it was first discovered, which is C. elegans, a worm, have now found 19 genes that govern that process. 11 of them are about cleaning up the mess, scavenging. For every gene they find in the worm, they find 5 to 10 invertebrates. Now that's a lot of genes, a lot of potential mutations. So imagine that somebody has a mutation, for example, in a scavenger receptor that is normally on cells that scavenge other cells that die normal programmed cell death. So an example I gave 12 years ago at a talk in Germany, when you make an immune response to flu, let's say flu, the B cells making antibodies go to a particular site called germinal centers. And there they multiply, they spew out 2,000 antibody molecules per second each, and they mutate. And they hypermutate. They mutate so much in order to get a few B cells that can have a better ability to bind to flu. Now, in the process of that mutation, a lot of B cells die. And there are special scavengers to pick them up. There's so much death there that there's special scavengers. They're called tingible body macrophages. Tingible because they're stainable, and stainable because they're so full of dying B cells. And I suggested, what's the possibility that somebody somewhere might have a mutation in the scavenger receptor that the tingible body macrophage uses to pick up the dying B cells? This is a person who would have a problem every time they had a B cell response. They would get flu, the B cells would start to respond, they would mutate, they would die, they wouldn't get scavenged, and so they would go into secondary necrosis. And they would release alarm signals and DNA, RNA, tubulin, SNRPs, all those molecules that you make antibodies to in lupus. This is a person who would flare every time they got an infection. And so people say to me, well, why is lupus so predominantly a female disease? And here again is the details. Post-puberty onset lupus has a female bias. Pre-puberty onset lupus does not. Now, that's a kind of hint. So here's my hand-waving answer. There's death that goes on in female bodies that doesn't go on in male bodies. We pop an ovary every month. That's not programmed cell death. That's a lot of scavenging. We kill off, clean up, and regenerate an entire uterine lining every month. That's a lot of scavenging. That's a lot of genetics. That's a lot of potential for mutation. So I suggest that one of the reasons there's a female bias in lupus is that there's a female bias in death and death genes and their process. And I was once talking, and a woman came up to me, she must have been in her 80s, and said she got lupus when she was 13. And her family physician, can you imagine how long ago this was? Her family physician told her that if she survived to menopause, it would probably go away. He knew that long ago. And she did survive to menopause, and it went away. So I have suggested to rheumatologists that when they get a female lupus patient right around puberty or late teens, early 20s, put them on the pill and don't let them cycle and see what happens. Don't let them cycle for a year or two. And they say, oh, we can't do that. That's not physiological. And I say, you know what? When I was a teenager, I was so fed up with that particular aspect of my own physiology that I went to 2 different family doctors. This is before you had to have parental consent. And I got pills from both of them. And I kept myself non-cycling for 4 years. And I'm still here. I don't have lupus. It's a lot better than dying because your kidneys fail. Okay, that's the 3rd category. The 4th category is a subset of this. It was invented by Tony and Livia Rosen at Hopkins. 1 minute. And they say the immune system is not involved, and their disease is scleroderma. And what they have found, to make a long story short, is that scleroderma is not an autoimmune disease. It's a disease that causes damage, might have something to do with heavy metal poisoning, and the immune system is responding to the damage. They call it immune impressionism. They say the immune system is painting a picture because the antibodies lead you to the right place, but it's not caused by the immune response. So in these 4 categories, there's nothing wrong with the immune response. It's either responding to an infection or to bad death and alarm signals or to damage. In the 5th category, there is something wrong. You make the wrong class. Remember I said that you make different class of immunity to catch worms than you do to catch viruses. You also make a different class of immunity in different tissues. If you have an immune response necessary in the gut, you make an antibody called IgA, which is secreted in the mucosa. If you have an immune response in the skin, you make what's called delayed type hypersensitivity, which is the response you make to poison ivy. The wrong kind of response in the wrong tissue can do damage even if it's not autoimmune. So what I'm saying here is if you start asking the right questions, you may be able to find the right answers. And I think that what's been gone wrong with the field of autoimmunity is we've been asking the wrong question. I have no prescription here. I can't say, like with transplant, block the alarm signals. But what I can say is if we start asking the right question, we might start having the right answers. So hopefully some of that impinges on what you do. Hopefully some of you will either come up and argue with me enough or have been convinced enough that perhaps we should start thinking about damage as something that turns on an immune response, and that we actually have ways of stopping it. They're not perfect yet, but we're getting there. The people in the transplant community are working very hard at it. And I think if we go there and learn some of what they're doing, we may also be able to use some of that for the immune responses that are messing up your critical care patients. So thank you very much for listening.